we're gonna develop new tools to detect Parkinson's and Alzheimer's disease early. I'm Tal Gilboa. I'm a postdoctoral fellow at Harvard at the Wyss Institute. And my team and I, during COVID, developed antibody and antigen tests, which we set up in a CLIA lab, and they got FDA approved and were commercialized with a big company. And now we're aiming to use all the knowledge we have to develop quantitative blood tests for neurological disorders, which involve aggregation of proteins, and specifically we'll start with Alzheimer's and Parkinson's disease, which is a huge market on its own. So currently when a person has initial symptoms for Parkinson's disease, he will go through a series of physical exams with the PCP. He will see a neurologist. They will require sometimes some very expensive imaging, MRI, PET scans, until they reach a subjective clinical diagnosis, because there are no tests currently available to diagnose Parkinson's disease. And this takes up to 2 years until they start treating these patients to minimize symptoms and slow down the progression of the diseases. And why we cannot detect the diseases early is because the brain of living patients is not accessible. We can't just biopsy the brain. And if we could, we would see that neurons are dying over time. So we have this loss of neurons, which occurs over time, but the actual initial event which triggered these diseases starts decades before symptom onset. And it happens really slow. Neurons are dying very slow. And so we have this very precious window for intervention if only we could diagnose these patients early enough. And this happens to millions of people globally, and more than 6 million people in the USA, which are diagnosed mostly with Alzheimer's and Parkinson's disease, resulting, here comes a number, Adam, with a huge economic burden to the payers and the healthcare system. But we do know that in very early stages, these patients secrete signals from the brain to the blood. So we have proteins, DNA, RNA, and also these very interesting vesicles, which are secreted from neurons. And these vesicles contain inside of them proteins from the cell of origin, for example, neurons, and they preserve them in the original context. And so we aim to use those molecules to open a window to the brain by developing a platform which will allow us to analyze molecularly the brain of living patients. And the way we're doing that is we have 2 different technologies which are complementary. One is for the isolation of these vesicles. This is, here you can see example of how they look like. It's these 2 circles. Yeah, sorry. 2 hands, 2 things, too many things. So we have methods we developed to isolate these vesicles from blood in an automated high throughput way. And we can take them, burst them open, and detect proteins at the single molecule level. My field is called single molecule biophysics. We're developing very sensitive sensors to detect these pathological proteins. So here you can see an example of how we can quantify these green dots, where each dot represents one aggregated protein in Parkinson's disease. And, oh wow, a lot. So confusing. Our go-to-market strategy is, first, we can use the device we already built for biomarker discovery as a service. We can just sell the device and do this automated pre-processing of blood samples. Our 2-year plan is to enable clinical trials. And there we can use it to screen for drugs and see how they interact or engage into the target. We can allow them to have specific biomarkers for patient selection. So it's very important. Now, many of you probably know, there are new drugs approved for Alzheimer's disease, and the reason they passed the clinical trial was because the patient selection was very accurate and was done with CSF samples. We will do it in blood. Our test will allow us to replace the very expensive amyloid PET scans or MRI, and to find stopping criteria for the new drugs that are developed, and to monitor disease progression. And our long-term plan is to have this early detection tool to allow us already during wellness visits to identify these patients and actually have a better cohort for the clinical trial to find new treatments for these diseases. And we are already doing that. We're partnering with AZI, which just released and got their first FDA approval. And actually, it's a huge market. There are many companies now doing clinical trials for these diseases. And for just more numbers, so we have more than 170 ongoing trials for Alzheimer's disease and 140 for Parkinson's disease. So we can really already partner with these ongoing clinical trials. And our 3-year plan is to first start with continue the R&D, of course, set up the service lab, and do POC with samples we already have. We're partnering with 2 biobanks at UPenn and in Brigham and Women's Hospital. We'll then continue to partner with clinical trials. Hopefully, we'll get some publications, but for the product, and expand to new customers. And our team brings lots of technological expertise. I'm a biomedical engineer by training, with 15 years of experience in the field, and specifically with sensors and diagnostics. Dima, my partner, he's an RNA biologist with lots of exosome experience over 10 years. And Maya is an MD PhD and a neuroscientist. The 3 of us are working together in the last 3.5 years. And we are in the lab of David R. Walt, which funded companies like Illumina and Quanterix. And we have also support from Private Foundation for the Alzheimer's disease. We're working with Chan Zuckerberg and Open Philanthropy, and for Parkinson's with the Michael J. Fox Foundation. We're raising money. We are hoping to get $5 million for the first 3 years. And I would love to speak with any of you and talk about partnerships and other opportunities. And here's my email. And thank you, Sai.